So, very good morning, good afternoon, good evening, everyone. So, as we have started a lecture series on network pharmacological approaches, and in earlier videos, just the introductions, we have what we are going to cover in network pharmacological approaches. We have discussed, if you have gone through that videos. So, what actually we are going to cover, we have discussed it. So today's in this series, I am going to discuss first part, that is the introductions about network pharmacology as well as system biology. So system biology is very important with respect to network pharmacology. First, there is a system biology, and network pharmacology is the one of the part of system biology. So first, you have to understand what actually the system biology is. So system biology, sorry. So you can see that it's a interdisciplinary field that studies biological system as a whole, not just the individual part. So when you are studying the whole system together, that comes under your system biology. In simpler word, you can see that we are not looking at one gene, one protein, one pathway in system biology, but system biologists, what they are doing is they are analyzing how all these components interact together to produce particular functions, particular behavior, or particular disease, right? So means we are not looking for individual what happens with particular gene, what actually happens with particular proteins, what is happening with particular pathways, but we are integrating all these genes, protein, metabolomics, all these together, and so help to understand what actually happens in physiological functions, in behavior, or if in the disease development as well. And we all know that when we talk about any disease is not caused by a single gene. There are multiple proteins, multiple factors which are involved in the development of disease. So system biology is again, it's a means in that one we are studying the biological systems as a whole, not as a individual part. So if you see that system biology are combining all these areas, when we talk about genomics, that means we are studying that particular particular genes, particular genomes, that comes under your genomics. At gene level, whatever we are doing the studies at the gene level, that comes under genomics. And you all knows DNAs are goes under the process of transcriptions and results in RNA. And when we are doing the study at the RNA levels, that we call transcriptomics. And RNAs are again goes under the process of translations and going the productions of protein. And when we are studying at the protein levels, that we call proteomics. And again, after that, there is metabolites which are formed. And when we are doing the study at the metabolite levels, that we call metabolomics, right? So these all, so genomics, transcriptomics, proteomics, metabolomics, epigenomics, these together, when we are studying that, we call system biology. So system biology combines all genomics, transcriptomics, proteomics, metabolomic, epigenomics. Epigenomics is the study of changes in the gene expressions and that occur without altering the DNA sequence. So whatever the changes you can see in the gene expressions, but without altering the DNA sequence, that we called epigenomics. And I hope many of you knows that the well-known examples with respect to epigenomics are DNA methylations, histone modifications, so, and each of which alter how gene are expressed without altering the underlying DNA sequence. That is your epigenomics, right? How your environmental factors are altering the genes levels. So when we are studying these in system biology, we are not studying these at individual levels, but combine we are combine these all together and try to understand what actually happens in particular disease or what actually happens in particular biology, that we call system biology. So system biology combines all these. So you can see that there is a network of cellulars, molecular network, gene network. So there are a lot of networks, and when you are studying it combined together, that is system biology in simpler words, right? So to study the system biology, so you should have a knowledge about computational methods, because we are using computational technique to understand all these networks. You should have a good understandings about the biology, to understand what actually the basic biological signalings are. And of course there is a new technologies, like you are creating a models, and that models will help us to predict what actually happening in the system at the system levels. So there, if you see that in system biologies, there is a you are generating a hypothesis, new insights, you are doing the analysis of genomics data, transcriptomics data, your metabolomic data, all these combined together we called omic technologies, right? So all these analysis we are doing, and to understand particularly so particular disease, particular physiology, particular metabolomic pathways, or all these together we are going to understand that. So that is the purpose of your system biology. So of course when we talk about system biology, you might have gone through some books on the system biology. You will find that mathematics I am not saying there is a very complex mathematics is involved in the system biology, but of course simple differential equations or simple mathematics, you should have understandings about mathematics as well. So you can see the example of simple mathematics, but lecture by lecture, when we'll go in the next lecture, next lectures, I will try to explain these mathematical equations in simpler way. So you can see on your screen, there is a, particularly for example, there is a protein A and it is changed to protein B. So if you want to calculate the rate of change of these proteins, so you can do it by applying the simple differential equations. So simple differential equations, you can do dA by dt. That is a change in the particular level of these proteins. Obviously, it will be the productions minus degradations. So this is very, very simple examples, for the mathematical equations which we are applying in the system biologies or in network pharmacology. So we are creating a model based on the informations which we have, and informations whatever we have on the basis of experimental data. And by applying these experimental data, we are creating a model, and we are validating this model, and that model will predict the futures. So but before model, after model creations, we have to be do the simulations. We are giving the simulations, and we check the validity of the models, and then we can predict the futures. So you can see that A to B, it can be, so both side A to B and B to A, if for examples, then you want to see the change in the concentrations of A. So by simple differential equations, dA by dt is equal to minus K1 A. The reason why there is a negative sign, because this is a constant, K1 and K2 are the constant. So A concentrations, because A is going to degrade and going to form the B. So of course the concentration of the A is going to decrease. So this negative represents your concentration is going to decrease when your A is converting into the B. For example, so your A protein is going to activate B proteins, and so its concentration is going to decrease. So that's why this negative sign represent that concentration of A is going to decrease. Plus K2, this is a constant and this is B, right? If as I said, in biological network, the things can go in both direction as well. A can activate B, and B can also go in the back directions. So if B is going to be A, so in that case, your rate of change will be dB by dt is equal to K1A. But in this case, your negative sign will be in this one, minus K2B. And why negative is here instead of here? The reason is because now your B is converting into the A, so concentration of B is going to decrease and A is in the positive side. So that is the rate of change in the concentration of B, and this equation is representing rate of change of the concentrations of A, right? So this is I am just added means not very complex equations, but just a basic understanding how we can create a models by simple differential equations. And as I said, after creations of the models, you have to give the simulations. and then you can use these models to understand the biology with respect to systems, right? So, when we talk about drugs, because I am a pharmacologist, so we always talk about the drugs and we talk about network pharmacology. So, this is a system biology, and as I said, network pharmacology is the part of system biology, because in biology, there are a number of things which are involved in biology, right? Biology contains number of other discipline. So, when we talk about pharmacology, pharmacology is specifically when we talk about study of the drugs which are there in the market and which are under preclinical studies, which are at discovery state, whatever they are. So, if you see that the drugs which are available in the market, they are based on single drug and single target concept. So, for examples, so you have a drug for the cardiovascular disorders. For example, the drug is prazosin. So, prazosin is going to block your alpha 1 receptors. So, that means your drug is going to block particular targets in the bodies and going to manage that disease. So, so what problem is, and there are many drugs are there based on this concept. But now we came to understand that the drug molecule is not going to interact with single targets. Whenever you took any drugs, is it going to interact with single target? No, it's going to interact with multiple targets, right? So when how drug is interacting with the multiple target, that is your network pharmacology. So if you see our animal models are also have a limitations, and they can only mimic symptoms, but without any evidence. What is the mechanism causing that symptoms in the animal model, whether that matches with the human disease or not, we don't understand that. And so that's why we have many drugs in the market, but the allopathic medicines are always criticized that these medicines are not doing the cure of the disease. Now question arise, why we are unable to cure the disease in most of the cases, why we are not able to do that in spite of we have a lot of advanced instrumentations. We have a lot of understandings about the things at the molecular levels and so on. The answer to this question is, one of the answer is, in most of the disease we don't know the etiology, we don't know what is the exact reason for that disease. For example, if we talk about hypertensions, and particularly primary hypertensions, 95% cases are of primary hypertension, remaining 5 are secondary hypertensions. And primary hypertension means where we don't know the exact reason of the hypertension. So when we don't know the exact reasons, so you can understand that can we have a drugs which can cure the disease? No, because we don't know the exact etiology. So whatever the drugs which are available in the market for the management of hypertensions, these are the drugs which are providing you the symptomatic relief, which is going to manage your blood pressures within normal range. And if you skip the medicines, again your blood pressure is going to increase. Similarly for diabetes, similarly for other diseases, if you are not taking the medicines, if you skipping the medicines, again your parameters is going to increase and going to damage, right? So that means there is a need to understand what is the exact etiology of the disease, so that we should have a molecule which will hit that etiology. So network pharmacology is the one of the area which can help us to understand the disease at the root levels, so that we can design a molecules for that. So network pharmacology, we all knows what actually the pharmacology is devoted to understand drug pharmacological mechanism in a network perspective. So as I said, your molecule is not going to interact with single target, but it's going to interact with multiple target, and it will create the network. That is the network means. So network pharmacology is a emerging discipline useful in drug discovery, and it combines genomic technology, system biology, chemistry through high performance computational tool. So it's a new frontier discipline of systematic drug research in the era of AI, and year by years you will get various analytical tool to in this field, and it globally regarded as a next generation medicine research mode. So, if we talk about biological network, human body at the steady state, at the natural equilibrium, is at the healthy state. So what are the topological parameters when the persons who have not have a disease, that is the healthy persons, what is the network? Because network is also there when person is in the healthy state. You are taking the foods, there is a lot of interaction is happening among the molecules, right? But it's a natural equilibrium state in the healthy state, and you are creating a stable network. And that stable network have specific topological structures and certain network properties, right? And whenever there is any pathological conditions, any disease are developing, so these topological, these network is going to disturb. So it could result in a unstable network. And when your network is unstable, you will get different topological structures and different network properties. So network pharmacology is particularly proposed based on the theory of network biology and biological balance. We all knows, means, there are a lot of endogenous molecules, they are interacting with a lot of targets, and we are getting the particular biological responses, and that is required for normal physiological functions. But when these are altered, particularly when we have any disease or disease which is going to develop, so these network is going to be, the stable equilibrium states are going to be disturbed. And by understanding this, what actually changes are happening, what are the changes in the networks is happening, we can thought of drugs, we can identify the promising drug molecules. So as I said, in network pharmacology, there are two things. One is pharmacology, another one is network. So I hope you got to know that what actually the network in simple terms. Network means, so you have a networking. If someone have a lot of networking, so that means you are connected with one to another, another to another, then another person is another network, another person have another network. So combinedly, when we study it, that is we call network, study of the network. So, what another terms which are there in the network is pharmacology. So, pharmacology, in simpler word, it's a study of drugs. But you know that when drugs reach at its target site, uh, if you, for example, you have a headache, you took the XYZ drug, and your headache has been resolved. So what there are two things when drug is acting on a target. One is drug action and another one is drug effect. So whatever we are observing clinically, that is we call drug effect. That you took the medicines, your headache has been resolved. That is a drug effect. What behind that, how that headache has been resolved, there is a lot of molecular, biochemical signaling pathways which are involved. Your drug is going to interact with that, first how you got the headache, because there are a lot of intracellular signaling pathways which has been changed, which are stimulated, inhibited, and ultimately the effect which you are getting, that is a visible effect, that we call drug, that we call effect which is visible, right? And how you have treated, so whatever your drug molecules have alter that signaling pathways, and ultimately you got the effect. So whatever the intracellular signaling pathways are happening, that is obviously these are the invisible. So when we talk about pharmacology, we always talk about these four. Most of the, means, approximately 80% or more than 80% of the drugs are showing their response through interactions with the receptors. There are some other targets as well, like non-receptor targets, like enzymes that can produce the effect by interacting with the enzymes. There are drugs which are available in the markets. The drugs can neutralize the acid like antacids. So, but most of the drugs, as I said, 80% of the drugs are showing their response by interactions through a receptors. So what are receptors? Receptors are the proteins, right? So these are the four major category of the receptors. One is nuclear receptors, another one is receptors, kinase, or also we called it a enzyme kinase linked receptors. There is a ligand-gated ion channel receptors. There is a GPCR. These all are membrane bound receptors. That means these all receptors are present on the cell membrane except nuclear receptors. Nuclear receptor is present inside the cells. So if you want to target the nuclear receptors, so your molecule should have the ability to cross the cell membrane. If your molecule is not able to cross the cell membrane, so how it will be interact with the nuclear receptors? So nuclear receptor is present intracellular. Intracellular means inside the cells, and whereas other these receptors are present on the membrane. Then effector in this case is nuclear receptor is gene transcriptions. So whenever your molecule is going to interact with the nuclear receptors, it's going to do the transcriptions, translations, and do the formations of the proteins. Whereas in kinase receptors, your effector is protein kinases. In ion channels, there is a ligand-gated ion channels. There is a effector is your ion channels, and here in GPCR, your channels as well as the enzymes. So a nuclear receptor is coupling with DNA, whereas this is a direct enzyme kinase receptors are not linking with another things. But G proteins also coupled in intracellular domains with the G protein. That's why we called it a G protein coupled receptor, because G protein is coupled inside the intracellular domain. Example, as I said, steroids are showing their response through nuclear receptors. Insulin, growth factors, cytokines, they are showing their response by interacting the enzyme kinase linked receptors. Ligand-gated ion channel receptors, acetylcholine in earlier videos also we have discussed many times. Acetylcholine is have a two receptors all over the body. One is muscarinic, another one is nicotinic. So nicotinic receptors particularly are your ligand-gated ion channel receptors, whereas muscarinic receptors are your GPCR. So there is a GABA, that is a gamma aminobutyric acid, that is the inhibitory neurotransmitters. So GABA A receptors are your also ligand-gated ion channel receptors. Adrenoreceptors, all the adrenoreceptors, alpha and beta, alpha 1, alpha 2, beta 1, beta 2, beta 3, all are your GPCR. So structures, we will discuss in detail about their structures with suitable diagrams to understand that what are their different structures and how intracellular signaling pathways are going to activate and how you are getting the effect. And why this is important with respect to network pharmacology, because without these basic understandings, you can't become a good network pharmacologist. So to become a good network pharmacologist, what is required? You should have a good understandings about the molecular pharmacology. You should have a good understandings about your system biology, little mathematical equations, and your computational techniques. You should be familiar with the computational techniques so that you can create a better model. You can learn the R package or other packages as well to become a good network pharmacologist. So in next video, we will discuss in detail about these receptors, how these intracellular signaling pathways is going to be activate. Uh, if you have any questions, you can put in the comment box, or if you have any suggestions, you can suggest it in the comment box so that in the next videos we can incorporate it. But I hope in conclusions in today's session is system biology, where you are not studying the things at individual levels like we are doing in the genomic, transcriptomics, proteomics, metabolomics, epigenomics. We are studying it combined together. And of course, when we are combining all these together, complexity will be there. So that's why these high performance computational techniques will help us to resolve that complexities, right? So this is all about this is the models simple differential equations. So thank you so much. Thank you.